Good morning and welcome back to my channel. So today we are starting lecture number 58, bio section. Title is mutation. So specifically we will be talking about point mutation, and the category of human disorder which we will study is sickle cell anemia. So see, there are a lot of hints in this name. First of all, anemia, it stands for a condition in which there is decreased level of RBC and decreased levels of hemoglobin in body. And second, the shape of RBC, the normal shape, healthy shape of RBC is biconcave. The biconcave shape, it increases the surface area, therefore efficient gas exchange is possible by RBC. But due to mutation in the hemoglobin beta chain, the shape changes and it becomes sickle shaped. And this in turn affects the quality, the life of RBC, and it causes cell rupture and therefore it leads to anemia. So let's look into the details of the genetic disorder and how it translates into a pathological condition. I have picked up the important points and summarized here. So this paragraph talks about mutation. So mutations are categorized into two broad categories: mutations in DNA sequence and mutation in chromosome structure. So in case of DNA sequences, these are small mutations. Okay, point mutation is one single base pair change, and it doesn't change the reading frame, but it changes the encoded amino acid, and therefore in turn it affects the protein structure and therefore function. So today we will only focus on point mutation. So see, this is the DNA sequence, this is the coding strand. So GAG sequence, it is mutated and this A is replaced by T, and this in turn changes the sequence of the complementary RNA. So see, this is the regular GAG and it changes to GUG. So if you check the codon table which we studied in molecular basis of inheritance, GAG codes for glutamic acid. Glutamic acid is negatively charged amino acid, and GUG codes for valine. This is non-polar amino acid. So if a negatively charged amino acid is replaced by non-polar, so imagine the kind of distortion it will produce in the structure of the protein. Okay, because this negatively charged amino acid, it definitely pairs up by electrostatic force of attraction to some positive areas in the globin chain. Now, if it is replaced by non-polar amino acid, this will open up the structure and therefore the distortion is seen. Okay, so this is an excellent example of how the genetic disorder manifests in the structure of protein. So point mutation arises due to change in single base pair of DNA. The classical example is sickle cell anemia. So what you have to remember is the glutamic acid at position number 6 is replaced by valine, and this is the mutation. GAG changes to GTG in the coding strand, and this code is for the beta chain of hemoglobin. Remember one thing, hemoglobin has 4 polypeptides, 2 alpha chains and 2 beta chains. So it is the beta chain which is affected in sickle cell anemia. So see, this is the structure of healthy RBC. It is biconcave, and this is the sickle shaped structure of sickle cell hemoglobin. So this abnormal hemoglobin is denoted as HbS. So under hypoxic condition, in deoxygenated state, it polymerizes into rigid fibers, and those rigid fibers, they cause extension of the cell membrane. It distorts the RBC into sickle shape, causing them to become inflexible, and this leads to all these problems: blood vessel blockages, hemolysis, because this structure will put pressure on the plasma membrane and it will cause rupture of RBC. So hemolysis, this in turn leads to anemia, chronic anemia, and also organ damage. So sickle cell anemia is autosomal linked recessive trait that can be transmitted from parents to offspring when both the partners are carriers for the genes. The disease is controlled by single pair of allele HBA and HBS. So we will look into the genotypes. So see, for hemoglobin, three different genotypes are possible. In homozygous individual for HbA, this is healthy condition. The healthy hemoglobin is denoted this way. The homozygous individual for HbS, they are denoted this way, and it shows diseased phenotype. So this is the actual genotype of sickle cell disease. And when you have heterozygous condition, one HbA and other HbS, these individuals, they apparently are unaffected, but they are carrier of the disease, as there is 50% probability of transmission of the mutant gene to the progeny. Homozygous condition is called sickle cell disease, and this heterozygous condition is called sickle cell trait. Keep this in mind. So see, everything in this slide is important. You should remember the substitution of amino acid, which amino acid is replaced. Glutamic acid is replaced by valine. What is the position, which chain of hemoglobin, and then what happens due to this mutation. The effect on hemoglobin. It undergoes polymerization when under low oxygen tension, and this causes change in the shape of RBC. So from biconcave disc to elongated sickle-like structure. So there are a lot of points. Make a note of it. So mutation generating factors. They can be physical or chemical. Those are called mutagens. And the example given here is UV radiation. It can cause mutation in organisms. So based on this concept, there is one question. Mutation in plant cells can be induced by. So zeatin and kinetin, these are two examples of cytokinin. This is naturally occurring cytokinin. This is synthetic cytokinin. So they are plant hormones. They will not cause mutation. Infrared rays are not mutagenic. Gamma rays are ionizing radiations. They are mutagenic. They changes the DNA structure. So option 4 is the answer. Next question, select the correct match. Sickle cell anemia, autosomal recessive trait, chromosome 11. So this part is correct. This part is not given in NCERT. So you can add this in additional information in your notes. The gene for beta chain is located on chromosome 11 and genes for alpha chain is on chromosome number 16. So this is correct. This is correct match. Thalassemia we haven't discussed yet. Hemophilia also we have not discussed. Phenylketonuria, autosomal dominant? No, it is autosomal recessive trait. It is not given in NCERT, but this was asked in NEET exam. Sickle cell anemia is due to quantitative problem of globin molecules. So see, it is the quality of globin which is affected in sickle cell anemia. The quantity is not affected. There is no reduction in synthesis of globin chains. So this statement is incorrect. Sickle cell anemia is a qualitative problem of globin molecules. Okay, quantitative problem is incorrect statement. The answer is thalassemia. When we discuss thalassemia, then you will be able to understand what this particular point. So with this, we have completed this section also, mutation sickle cell anemia. I want to remind you this that it was not mentioned in NTA syllabus. I just discussed this topic because it is mentioned in NCERT. So Monday we will discuss this topic, sex determination in humans, birds and honeybee. Thanks a lot for watching this video and take good care of yourself and enjoy this weekend.